I'd like to let's welcome Zoe and hear what she has to say. Thank you.
Thank you, Roy. All right. Thank you so much. It's really an honor to be here. I want to say thank you for everybody for coming out on a Tuesday evening and spending it with me. So Roy introduced me, but I thought I would start by giving you a little bit of a snapshot of something that happens frequently in my life. So it's not uncommon for me to find myself in social situations, especially with non-scientists, and they ask me, what do you do? And I say neuroscience. The first thing that happens is they look for anyone else to talk to. But then they realize they're stuck with me and they ask me, what do I study? And so this is where the conversation often takes a turning point, because I say I study monogamy. And this inevitably leads to the question, are humans actually monogamous? So this is where I get to put on that hat that they give you when you get your PhD and give this very eloquent answer of sort of and sometimes. And this is because when it comes to humans, we're actually really incredible at showing a diversity of different mating strategies. And so as you can see in this picture here, classically, we've engaged in all sorts of different family units. So on the bottom would be considered what would be normative for the Western world, where you have mom and dad and the kids. But we also have parts of the world where it's normal for one husband to have multiple wives. And although much rarer, it also occurs that you can have one wife with multiple husbands. On the top right here, what you see is basically a small village in Nepal where all of the brothers marry the same woman. Now, there's many reasons why we engage in these different strategies, but some of these are very human-specific and involve things like religion and culture. But the thing about all of this is that we do form these units, and they're essentially held together by social bonds. So regardless of our variation in mating strategies, we have this amazing capacity to form long-term bonds with each other. And these bonds are incredibly important for our health. If you have a healthy social life and healthy bonds, friendships, spousal relationships, you literally live longer. And if you are lonely, that is a mortality risk factor that's equivalent to smoking cigarettes or being obese. So there are many good health-related reasons why we study these bonds, but if I'm being totally honest with you, the reason that I study these bonds is because they can be completely irrational. They make us do absolutely astounding things. They have changed the course of human history. And so I want to understand these bonds, and particularly I want to understand where do they live in our biology. So you might be asking yourself, well, great, so how do you do this? One way we can do this is by studying humans. And this is actually great because you can ask humans questions. You can say, how much do you miss your partner when you're away from them? How much do you fight with each other? You can give them a little test that are two little circles, and you can say, you're this circle, your partner's this circle. How much overlap do you see in yourselves? But here's the thing. I want to understand these bonds in our biology, which means that we need to get down to the cellular and molecular origins of this. And unfortunately, we can't do this in humans because we don't have the technology that lets us go in and peer into human brains. So what this means is that we need some animal that can serve as a proxy for these complex social behaviors that we have. And as soon as I say animal, you probably think to yourself, laboratory mice. And you would be right, because these animals are the workhorse of biomedicine. And this is obviously a mouse, this street art, because notice the pointy nose and the long tail. But there's one problem with this. If we're going to study bonding in the laboratory, it means we need a laboratory species that's actually capable of forming long-term bonds, and that's not mice, because these guys are not monogamous, and they don't form long-term relationships with other mice in adulthood. Fortunately for me, though, there is a laboratory amenable species, a little rodent that's about tennis ball size, that does form these long-term bonds. This is obviously a prairie vole. You can tell because it has a little rounded snout and a little tail. And unlike mice, prairie voles are monogamous, meaning that they form long-term partnerships that can last their entire lifetime between a male and a female. We've actually, as scientists, been studying prairie voles for many, many decades, which has led to plenty of headlines in scientific journals, but it has also led to things like self-help books. So here you can see one that Make Love Like a Prairie Vole, 6 Steps to Passionate, Plentiful, and Monogamous Sex. presumably in an underground burrow. So you might be asking yourself, how do we take something like monogamy and social bonds? We're incredibly complex, and if we wanted to study them in humans, we would simply ask people, but we can't do that with an animal. So what do we do instead? Well, what we do is we turn to a test that's been used by scientists for nearly 50 years. It's appropriately named the partner preference test because it simply asks an animal, who do you want to spend your time with? This is actually a very old video, so I apologize for the quality, but what you see here is an animal that can freely roam around both chambers and then two animals that are tethered on the sides of these. And the first thing you'll see is that in a moment here, we're going to zoom in, and the test animal actually gets into an agonistic interaction with this other individual. This is really, really common because when prairie voles form bonds with their partners, they're selectively affiliative towards their partner. In other words, they'll spend the vast majority of the time doing this huddling behavior. Now, as scientists, we call this huddling, presumably because cuddling is too anthropomorphic. But this test allows us to essentially ask, have these animals formed a bond? And we can do this test in different flavors. We can have a male choosing between two females or a female choosing between two males. But over the course of 3 hours, we simply measure who do you spend your time with. And what we see is that animals form a bond, basically spend a ton of time huddling with their partner and not very much time with the stranger. But if they haven't formed a bond, then on average, you won't see any distinction in the time that they spend with these two animals. Now, I want to highlight how unusual this behavior is, because for the vast majority of species in the world, they don't bond after they mate. Instead, what happens is something that we refer to as the Coolidge effect. This is actually named after President Coolidge, and it refers to a time when he and his wife were on a farm tour. And so his wife and the farmer were sitting outside of the chicken coop, and she sees a rooster who's copulating with a hen. And she turns to the farmer and she asks, does this happen frequently? And the farmer says, yes, ma'am, multiple times a day. And she says, make sure you tell my husband. So later, President Coolidge comes by, and the farmer does as he's told, and he says, President Coolidge, your wife wanted me to tell you that this rooster mates multiple times a day. Now, President Coolidge was a man of few words, so he turns to the farmer and he says, always with the same hen? And the farmer says, no, always with a different one. And President Coolidge said, make sure you tell my wife. So for the vast majority of species, this is normal behavior. If you've mated with an individual, then you should prioritize paying attention to a different individual. So this is dramatically different than what we see in a monogamous species such as prairie voles. So how do we actually go about using this behavioral test to try to understand the hormones that are involved in the formation of social bonds? And so what I'm going to tell you in the next part of the talk is about a hormone you may have heard of before. It's called oxytocin. It's a hormone that we give to women to induce labor, but it's actually involved in many other facets of reproduction. You need it for letdown, but you also wind up needing it to be able to form bonds between parents and offspring. And it was this latter role that ultimately inspired scientists to ask if oxytocin might also be involved in bonding in other types of bonds, such as pair bonds between mated individuals. So how do we actually do this? This is where we can take advantage of a unique facet of prairie voles, which is essentially that if they mate with their partner and then we do this test, we see the emergence of a partner preference telling us that they had a bond. But we can actually manipulate their systems just a little bit to make it so the female's not receptive and they won't mate with each other. So they're cohabitating and interacting, but they're not mating. And under this situation, they don't actually form a bond. So now what we can do is we can take oxytocin, this small molecule that your brain produces, and we can ask, is this enough to form a bond when they wouldn't normally? And we can take animals and we can block the mating, but we can infuse oxytocin into their brains. And when we do this, we see that it essentially does the same thing as mating. It stimulates the formation of this bond. Now we can do the converse experiment as well. We can create conditions where they would normally bond. We can block the signaling of this particular hormone in the brain. And when we do this, we see that it blocks the formation of these bonds. And so it's experiments like these that led scientists to say that oxytocin is not only necessary, but also sufficient for the formation of social bonds. Now, as you can imagine, work like this often leads to pretty salacious headlines, a couple of which are shown here. And so this is extrapolating what we do from voles and making assumptions about what's happening in humans. And so I think it's relevant to ask, what is oxytocin doing in humans? And it turns out that it's important for almost every aspect of our social cognition. And this is work that sort of was initiated by one paper with a very simple title, Oxytocin Increases Trust in Humans. So in this paper, what they did is they took intranasal oxytocin. I brought some today. This is from Amazon. Don't buy it from Amazon. They use it from a pharmacy in the study. And they had people actually sniff this. So they applied intranasal oxytocin, and then they asked people, how much money would you be willing to invest with a stranger? And they saw that if they gave intranasal to people, they were willing to invest more money than the placebo control group. And the take home from this is that oxytocin increases trust in humans. Now you can imagine all of the scenarios that sort of unfold from here. People like, oh no, what if they start using oxytocin like in the sales room of the car dealership? And the thing that always gets overlooked in this is that this would affect the seller just as much as it would affect the person that was purchasing the car. So I mentioned that this was sort of a watershed paper because it opened a floodgate of studying oxytocin in humans using this approach with intranasal oxytocin. And so subsequent findings revealed that oxytocin can do things like make us better at inferring the emotional state of others based on their facial expressions. There's another study I really like that showed that when you give intranasal oxytocin to men, if they're in a relationship, they stand farther away from an attractive woman that they don't know. But if they're not in a relationship, it has no effect, suggesting that there's this state-dependent function of oxytocin that helps you to maintain your relationships over time. And in one of my favorite studies ever, which was published about 10 years ago, they showed that oxytocin also helps us bond with our pets. Now, this study actually focused on dogs, and I just got back from a conference where the same authors were presenting work, and it turns out that this is not true if you're a cat owner, which I think is pretty characteristic of cat relationships, and I am a cat lover, so I can say that. All right, so this first part of the talk, you know, it's not love, it's oxytocin. I hopefully left you with the message that it's overly simplistic to distill our incredibly complex relationships down to a single hormone. That's absolutely essential, but definitely not the whole story. And so a few years ago, my laboratory said, instead of focusing on the hormones that you need to be able to form bonds, what do you actually need to keep this bond going over time? How do you maintain these bonds? And this is where it turns out that dopamine is really essential. So you've probably heard of dopamine. It's certainly no stranger to its own salacious headlines, as well as podcasters who are willing to sell you strategies and supplements to optimize your dopamine. You do not need to buy these. But for this part of the talk, what I thought I would do is sometimes with my lab, we like to think about what the popular press would do with our research. And so I've interspersed a couple of the more salacious headlines that we've come up with. So what is the role of dopamine in monogamy? Or as I'll refer to this part of the talk, it's not love, it's just dopamine. So scientists have studied dopamine for a really long time. And they've studied it in the context of reward, right? This is the whole idea, dopamine makes you feel good. But it turns out that what dopamine really does is it acts somewhere along this axis. So at the bottom of this axis is what we would refer to as liking. This is basically if you're sitting on the couch and you have your entire bag of potato chips, you might sit there and just finish the entire bag. This is because this is a consumatory behavior that doesn't take very much effort. But on the other end of this axis is things that we really want, that we're willing to expend a lot of effort seeking out to obtain. And so for a long time, we knew that you don't actually need dopamine to like things. You can completely get rid of dopamine in mice, and they'll still sit there at the sucrose port and just lick sugar all day. But you do need dopamine over here for these high-effort seeking behaviors. And so we started thinking about this. These seeking behaviors are absolutely central to maintaining bonds. You need liking, certainly. If you don't like your partner, that's kind of problematic. But if you don't have a desire to reunite with them when they're away from you, that's also not a good way to maintain a bond. And so we started thinking about sort of how voles might be selectively motivated to be with their partner. This is basically how you want to be with your partner instead of somebody that you don't know. And we thought about everything that had been done with prairie voles and realized that the partner preference test, it doesn't really tap into this partner-specific motivation, this desire to be with a partner. Just like if you put a box of Cheez-Its in front of me, it doesn't tell you how much I'm willing to work for those Cheez-Its, but I will eat the entire box. And so we started thinking, well, actually, this partner preference test belongs on this end of the quadrant. And that's because in order to get consume the reward of being with the partner, it's relatively low effort. They just wander over to their partner, they plop down and spend the whole test over here. So we need some way instead that we can access this effortful motivation to be with a partner. And so this is a picture of a vole climbing up a bank that's supposed to remind you of that person that was climbing that mountain. So how do we do this? Well, we do this the way scientists have done this for decades, by essentially asking voles to work to gain transient access to their monogamous partner. So this is our first headline. Scientists automate the vole cuddle dungeon. What does this mean for your love life? Okay, so what does this so-called vole cuddle dungeon actually look like? Well, this is where my lab teamed up with a couple of scientists as well as undergraduate and graduate level engineers from CU Boulder so that we could build this apparatus that you see here. And what you have here is a top-down view where we have our focal animal in the middle, and then we have the partner tethered on this side and the stranger on this side. And what you'll see is that there's two levers that come out here. And the vole can pick one, and it can press that lever, and then it has to wait 5 seconds before it actually gets the reward. And watching videos teaches you how long 5 seconds is. But at the end of it, it picked the lever for the partner, and so it gets to go hang out with its partner for a moment. Now in this task, under control conditions, we see that they'll press the lever a bunch of times for their partner, but if we block dopamine signaling, this basically goes away. They won't do it anymore. This doesn't affect their locomotion, it doesn't affect their coordination, it's just that the effort is not there. The motivation is gone. But what about, I told you that the partner preference test is the other end of the spectrum, and we don't expect to be dopamine dependent. And so we can do the exact same experiment in the exact same animals, but now instead of measuring effort to access the partner, we can measure partner preference. And when we do this, we see that dopamine is not required. Basically, we can block dopamine signaling, and their partner preference is completely intact. They still like their partner. And so what this tells us is that partner preference is like liking. You don't need dopamine for this, but you do need dopamine for this effort-filled, effort-driven behavior to reunite with your partner when they're not there. So if you were reviewing this paper, what you should say is, so what? You've told me what we already know. But this is ultimately a foundation for asking the question of how does dopamine actually make you want to be with your partner more than some other animal? And we reasoned that maybe what's happening is that there's a difference in dopamine release, where there's more release of dopamine when these animals are trying to get to their partner than when they're doing the exact same behavior to access a vole that they don't know. And so to test this hypothesis, we needed some way to be able to measure dopamine in real time with sub-second resolution in the brain of a behaving animal. And so the way that we do this is we combine advances in what I like to refer to as wetware and hardware. So on the wetware side, we take advantage of advances in molecular biology that give us a fluorescence sensor where fluorescence provides us with a readout of how much dopamine is present. So bear with me, the way that this works is that we have a dopamine receptor that's actually been fused to a fluorescent molecule. And when dopamine is absent, this fluorescent molecule is actually in a conformation that has really low levels of fluorescence, as indicated by this little squiggle right here. But when dopamine is present, it binds to this receptor, changes the shape of this protein, which in turn changes the shape of the fluorescent protein, and now a lot of light is coming out of that fluorescent protein. Now, dopamine can bind and unbind really rapidly, leading to a change in fluorescence output that is similarly rapid, such that as dopamine is released, fluorescence levels go up, and as dopamine is scavenged away from the synapse, the fluorescence level goes back down. So we have a way to produce this within the brain, produce the fluorescence, but we need a way to actually measure it. And so for this, what we do is we combine a laser with a fiber optic that actually reads out the fluorescence from this molecular tool in real time as the animal is running around. And so scientists use viral glowing dopamine and lasers to unravel the secrets of love. So what does this actually look like when we're doing this in our voles? So up here you can see just a diagram of the apparatus, our cuddle dungeon that I showed you. And in each trial, what happens is that the lever comes out, they can press it, the door opens, and then they can run over it and interact with another vole. And so what you're going to see on the bottom of the screen here is that actually the amount of dopamine that's being released as they're engaging in these behaviors. And I've split these lines into two colors, where the blue line is how much is being released when they're doing this task to access their partner. And the orange line is what's happening when they're doing this task, but they're getting access to a vole that they don't know. So the lever comes out, and then they can make this decision to run over and press the lever. So already we see a differentiation here, where there's more dopamine if they're running over to press the lever to gain access to their partner. Then, as I mentioned, there's this 5-second wait period. And here again, what we see is this increase in dopamine that's much greater if they're doing this to access their partner, almost like this is an anticipatory signal or about to reunite. Then that door opens, and we see the dopamine jump back up again. And you have more dopamine being released if it's for the partner than for an animal they don't know. Now, after all of this, they can actually go over and reunite with their partner. And when they do this, what we see here is a very clear signal that they also get a little bit of dopamine release when they interact with their partner, which they don't get when they interact with the stranger. And so we think that this enhanced dopamine release for the partner is not only driving their motivated behavior, making them want to work extra hard to get to their partner, but also serving as a reward signal once they reach that partner, which I think is really nicely summarized by the following GIF. All right, so here's the thing. As in humans, vole love doesn't always last. And so we next asked, what happens to this chemical signature of love in the brain when the bond is broken? So you might be asking yourself, how do you break a bond? And the answer is very simply, you take partners away from each other and you put them in separate rooms for a few weeks. And we did a whole time course, so we know that it takes about 4 weeks to break a bond in voles. And the way we know that is that's how long it takes before they can form a new bond with a new partner. And so we did that here, and this is just to remind you, this is what the dopamine looks like. There's a lot more of it for the partner when they're interacting before they're separated. But after 4 weeks of separation, from other work, we know they can still remember their partner, but now they're not actually getting that same increase in dopamine. If anything, they might be getting a little bit more when they interact with the stranger. So what we think that this means is that there was this, oh, sorry, I missed a headline. This is our last headline. Have you really moved on from your ex? Take our online dopamine test and find out. And as federal funding for research creators, this is how I will maintain my lab. So what's going on here? We think that basically over time, this sort of reward that's associated with the partner goes away. So now there's no distinction. In other words, the erosion of this enhanced partner dopamine may be what enables these animals to form a new bond. You could be saying, well, neato, cool, but why does this matter? This matters because it may ultimately help us identify ways to adapt to loss. All of us will lose someone in our lifetime, and it is the most traumatic thing that people report encountering, the most stressful event in life. And it is very, very difficult. And there's a subset of individuals that have difficulty adapting. And so one of the goals of my lab is to develop new strategies and interventions that help people adapt to the loss. Not forget it, but move forward in a meaningful way. Okay, so I've presented so far a hormone and a neurochemical that we know are really important for forming bonds and maintaining bonds, but it's not all hormones and chemicals. Hormones and chemicals act on your body, they act on your brain in order to shape your neural activity. And so the other line of research that my laboratory is focused on is basically saying that there might be something really important about this interaction that you have with your partner. And I bring this up because we know that relationships ultimately mature and become unique through a form of social learning. So if you think about your relationship with a friend or with a spouse, it's going to look different than the same type of relationship, spousal or whatnot, between another two people. So how does this actually happen? How do we have this unique connection with one person? Well, we can think of learning as this feedback loop where I might have some internal state that elicits a behavior I direct at someone else. They perceive it, it changes their internal state, and then they direct a behavior at me. So it's literally you learning about a partner, and this is where things can veer into friendly or unfriendly territory. But the thing is, this feedback loop between two individuals winds up occurring over and over again through shared experiences. And ultimately, what that leads to is very highly relationship-specific behavior. So this is my favorite one here. Right? So only the two of them do this. And it's akin to having like a secret handshake or just sort of like a habitual set of motions that you do with your partner. And it turns out that we can actually study the maturation of these relationships and the unique behaviors that emerge by looking at prairie voles, where we have the same sort of interaction loops. They have shared experiences, albeit in their cage, and they develop relationship-specific idiosyncrasies. And so what I'm going to show you in the next part of this talk is that we know now that bonding actually leads to synchrony with your partner at multiple different biological levels. So we know that just like humans, prairie vole bonds strengthen with time, and we do this just by doing a time course. So we take voles and we look at them before they have bonds, 2 days into bonding and 14 days into bonding. And what we see is that over this time course, they want to spend more time with their partner and less time with the novel vole that they don't know. So that provides evidence that there's maturation of these relationships. But what about the emergence of these relationship-specific behaviors? Do we have evidence that prairie voles, like us, organize their behavior with a partner? And so one of my favorite examples of organized behavior you can see here. But in prairie voles, we didn't teach them how to tango. But what we did do is we basically said, is there some evidence naturally that they organize their behavior as their relationships mature? And so what we did is we took animals, we paired them for 2 days, and then we did 2 reciprocal back-to-back partner preference tests. So we tested the male between his female partner and a female he didn't know, and we did the same exact thing, but now the female gets to choose, and we randomized this. And after 2 days, this is what the data look like. So let me take you through this. So this is how much time the males spend huddling with the partner. This is how much time the females spend huddling with their partner. And the line connecting the two is the pair. Now don't get too worried. There's really nothing here. But we put the animals back together for 2 weeks, and then we do the exact same thing. And now what we see looks pretty different. Because what this data tells us is that we have some pairs that really like to huddle with each other, and some pairs that spend a lot less time huddling with each other. But one of the things I find really striking about these data is it's incredibly organized, where the females huddle about 1.25 to 1.7 times as much as their male counterpart. Now, I just want to highlight, I love anecdata. There's one pair that doesn't do this, and it was the only pair that didn't successfully get pregnant. So this organization of behavior within a pair, which for multiple reasons we think is actually driven by the female, tells us that there is something unique to each one of these relationships. And so what are the potential mechanisms that could be contributing to this? So this is where we actually went back to humans, because it turns out that as humans, when we interact with other individuals, we actually wind up matching our brain waves together. And so this is what this little picture is meant to depict. So when these two individuals are facing apart, they have these brain waves and they don't look very similar. But when they're facing each other, they have these brain waves that are nearly identical. So brain waves are just patterns of oscillatory electrical activity in your brain. But the thing that really sort of drew us in is that when you look at interbrain synchrony, how close these wavelet patterns are, how strongly they're synchronized, is actually predicted by the strength of the relationship, such that we synchronize much more strongly with individuals that we're close to than individuals that we don't know. And so we thought, well, maybe, this also happens in prairie voles, but we don't know. So we started by asking, do we have any evidence of this interbrain synchrony in prairie voles? And so we measured the activity in the brains of 2 prairie voles that were either separated by a divider. We then take the divider out so they can interact, and then we put the divider back in. And so these animals, when the divider is in here, they have all the same room cues, they're in the same arena. And when we look at the neural activity of these 2 animals, what you see here, pink is female, blue is male, that when this divider is in here, there's not much concordance in these activity patterns. But when we take the divider out and they're interacting with each other, now these patterns start matching each other much more strongly, meaning that voles, just like us, have the ability to align their neural activity patterns when they're interacting with each other. So is this actually stronger with a bonded partner? And so here is basically insignificant neural synchrony when they're separated. We see that this goes up. They synchronize a little bit with a stranger, but when we give them their partner, this goes up quite a bit. So they do the same thing as us. They show stronger synchrony with their bonded partner. So we think that this is one real-time mechanism that can contribute to the feelings of connectedness that we have with a partner. But one of the things that fascinates me about our bonds is we can feel connected to somebody when we're not even in the same room with them. And so we think that this mechanism, which is highly dependent upon the actual interaction between two animals, is probably not the mechanism that's encoding this sense of connectedness that can last for a really long time. And so we stepped back and said, what kinds of mechanisms in our brains are really good at this sort of long-term change in behavior? And the answer that immediately leapt out to us is actually changes in gene expression. So gene expression within your brain leads to all sorts of things that happen on slow time scales. So gene expression in your brain is why you feel sleepy at night and awake during the day, but it can also be a harbinger of like large-scale shifts that can take a while to change, such as when you're depressed and you're coming out of a depression. And so we thought, well, this is not really about state change per se, but maybe what's happening is that these animals start with a particular gene expression landscape. When they're unbonded, this would be Vol1 and Vol2. Yes, we made these hearts because it's cute. And then when these animals bond, what happens is that both of them wind up with transcriptional states that are more similar to each other than to any other Vol. And so we did this experiment, and that is indeed what we found. But anytime you're working with gene expression, you want to know, what genes are underlying this? So we didn't really care what genes. We wanted to know, is there a subset of genes that is actually underlying this? And the answer is yes. So we looked through sort of 24 different sets of genes. I won't belabor the math. And we found one that's correlated between partners, meaning that when one animal has really high expression of these genes, their partner also has high expression of these genes. So we have this sort of convergence of the transcriptional landscape in two brains. There's a subset of genes that are actually synchronized in their expression between these brains. And so the question becomes, is this actually important for the emergence of those pairwise specific behaviors? So the way that we tackled this question is that we did it like the world's most simple behavior test. We take two voles that are bonded with each other, we take them out of their home cage, and we just throw them into an arena. They have food, they have water, and they have each other. And we measured how much time do you spend interacting with your partner. Turns out that there's tremendous variation where some pairs spend twice as much time interacting with each other as other pairs. And what we can do then is we can look at the expression of this subset of genes that's correlated between partners, and we can ask whether or not it predicts the pair's behavior. And the answer is yes. So what you see here is basically that if you have high levels of expression of these genes in both individuals, then you also spend more time interacting together as a pair. So we have a genetic correlate that can explain these pairwise differences in behavior. But this is something you could never understand if you looked at only one animal in the pair, because this is a behavior that necessarily requires two animals. So I sort of painted this picture at the beginning of the talk, where we have this innate ability to form bonds that's hard-coded in our DNA that sets us up with a brain that has this ability to form bonds. But what we actually know is that this is not a unidirectional process, because when we form bonds, there's these dramatic changes in behavior, and that's mediated by changes in our brain and also changes in our gene expression. And so this focus on pairwise behavior, I think, has really highlighted that we may have identified interbrain synchrony as one potential mediator of this relationship between behavior and brains, and transcriptional concordance as mediating the sort of long-term sense of connectedness that we have. Now, this is where I really like to speculate that these mechanisms that we can only measure if we look at two animals at once are ultimately what underlie the emergent properties of relationships. So by that, I mean the things that we can do only for the person that we're in a relationship with. So things like predict their behavior, share their emotional state, create a shared sense of self, or if you want to really go out on a limb, have a shared perception of reality. This kind of makes sense. If your brains are more similar, you might be interpreting the world in a similar way. And so the take-home message here is that we really need to study two brains at once to understand social behavior, which might sound completely straightforward, but has not been the typical approach in social neuroscience for as long as it's existed. Okay, I have one more part of this talk for you, and it's basically the question that you're required to answer if you're a scientist or you're in business, etc., right now at this moment in time, and it's what about AI, right? Okay, so we are deploying AI in my laboratory, and what we're doing is we're using it to get a really fine-grained sense of behavior within these animals. So we know that prairie voles can exhibit all sorts of complex behaviors. Here's an example of a dispute in a pair of prairie voles. So here, this is a male and a female that live together. We give them these little nestlets, and as you can see, they cannot agree on which side of the nest they should, or which side of the cage they should make this nest. But they're both very stubborn. So we have these behavior tests that I showed you where, we can ask about their preference for their partner. We can even ask how motivated they are to reunite. But that falls short of getting at these sort of natural behaviors that define relationships. And so we thought, how can we actually get at this level of resolution of behavior? And this is where we've been using AI-based tracking in our animals. So the way that we used to do this is that we may had an undergraduate or a graduate student in our lab sit there and watch videos frame by frame and sort of say, I think this is what the voles are doing. This takes hours and hours and hours. If you're having trouble sleeping at night, this is incredibly effective. But it's also subjective, right? It's some human saying, I think that this is the behavior that the vole is doing. And so when all of the new AI-based technologies started coming out, we said, maybe we can, this is what AI was built for, doing all the things that as humans we don't want to have to do. And so maybe we can train a computer to do this. And so that's exactly what we did. We started by basically training a computer to identify the different parts of the voles. And so now it can do that for an entire video. And so you can see that playing out right here. So this is step one. We just want to know where the different parts of the animals are through the entire video. From this, what we can then do is we can basically say there's a whole bunch of different features that comprise behaviors. So those are things like this, where the distance between voles, or whether they're facing each other or away from each other, and how quickly they're moving towards or away from each other. And so with those little dots, we can calculate all of these features. We can throw those features into an AI, and we can say, can you identify behaviors from what we just gave you? And in case you were wondering, this is what a computer thinks behavior is. So this big cloud that's been color-coded here is basically a series of dots where the computer says, this dot, this set of frames in the video, is basically the same as this set of frames that's next to it. So it's essentially assigning similarity scores to frames within the video based on the features. Now this is actually meaningful. There's 20 different colors in here, and those 20 are assigned to what the computer thinks is the same behavior, and it turns out that it's actually pretty good at this. Now poised to ask how these pair-specific behaviors can emerge as bonds mature, and we can ask questions like whether or not enhanced predictability of behavior contributes to relationship success. Now this is really important because we've all heard the term happy wife, happy life, but really it's happy bond, right? And we know that these bonds are so essential to our health that if we can kind of tap into how to make people happy in their relationships, this has really important implications for our health. Okay, so I have one more little section, and that's because I want to tell you a little bit about what we're doing next in my laboratory. And in order to tell you this story, I actually need to jump back in time. So this is me as an undergraduate when I thought I wanted to be a field ecologist. And so this is me in the rainforests of Nicaragua, where I was actually studying the social behavior of a very different creature, these guys. So you've probably come across this type of animal before, called a daddy longlegs or a harvestman. The scientific name is a pilionid. And basically these guys were notable because they would form these aggregations always on these spiny palm trees during the day, and then they would disperse at night. And this was fabulous. I loved it. But what I didn't love was the lack of control. Because you're in the rainforest, you can't control the weather, you have to get on your plane home whenever it's leaving, so you can't always repeat your experiments. I just didn't like the lack of control. And so for that reason, when I went to graduate school, I decided I was going to become a laboratory neuroscientist where we could control lots and lots of variables. But here's the thing, I've been that for 20 years now. And the realization that I've had along the way is that brains were not actually evolved to do these tasks we give voles, where we put them into these chambers and say, choose which partner you want. Like, I don't know about you, but I don't think I've ever had to choose my partner tethered on one wall versus a human on the other wall, right? So there's tremendous information to be gained here. But at the end of the day, brains have evolved so that we can navigate an incredibly complex natural world. And so I think that we need to be moving as neuroscientists in a direction where we can actually study brains in these environments, We have a lot of fundamental, foundational knowledge from the laboratory. Does this actually hold true in the wild? Now, I'm particularly privileged in the species that I study because there's actually a tremendous amount of information about these voles in the wild. And in fact, studying these voles in the wild is what led to the discovery that they're monogamous at all. And I'm just going to show you how that happened. So we actually have 2 species of voles in my lab. I've only talked about the monogamous prairie voles today, but we also have promiscuous meadow voles. These animals live in the same prairies. They can actually encounter each other in the wild. And so a field biologist that didn't actually care about prairie voles and monogamy was just out there trapping all the rodents that he could, and he would trap them in these little live traps, and then he would let them go again. But what he realized is as he was identifying these different vole species, this is just the number of paired captures, meaning 2 voles in the same trap, that you basically never found 2 voles in the same trap if it was a meadow vole, but that you often found 2 voles together in the same trap if it was a prairie vole. And so this observation led him to speculate that maybe these animals are monogamous and 2 animals are sharing a territory together. And so he reached out to a behavioral neuroendocrinologist and said, how could we study the hormonal basis of monogamy? And that's the origin story of the entire field that I'm in. So I think it's time for us to return to this type of setting, but doing so in a really smart way by deploying the same technologies that have made it so powerful to study the brain in the laboratory. And I think this is important because we've always had this trade-off where we sort of are in this box here where we can either have high interpretability or high ecological validity, but not both. And so traditionally in the laboratory, we have low ecological validity, but because we have so much experimental control, we have really high interpretability. I think we can actually continue this, keeping the high interpretability while gaining ecological validity if we're smart about how we do it. And so what my laboratory is working on is basically maintaining this window up here with high interpretability by doing this in a stepwise fashion, where we take what I've shown you today as findings from our laboratory setting, and now we test whether or not this is true in a more complex setting, using as an intermediate what I like to refer to as the volcosm. So this is just a room where we can let the voles roam free. And then we can take this and say, well, now that we've done this, can we move into a much more naturalistic setting? And does it still hold true in nature? And so this we do in something that we call Volterra, which I'll talk about in just a minute. This is obviously a huge undertaking, and so you might be saying to yourself, why are you even bothering with this? It's really expensive, it's hard to do. And there's many good reasons from a scientific perspective, but I think one of the most compelling reasons to do this from a human health perspective is actually that of all the drugs that go into phase 1 clinical trials, only about 10% actually make it to market. This is even lower if you look at the drugs that are behavior modifying, so antidepressants, etc. And part of this failure is because when we develop drugs, what we do is we go and we use them and we say, does this change the behavior of an inbred mouse in this highly restrictive laboratory setting? And then when we put it into a complex setting, it doesn't do the same thing, which is kind of unsurprising. We now have a pretty controlled way in which we can ask, do these drugs actually work if we increase the complexity of the setting to be a better mimic for the human situation? So what does this actually entail? So we have our volcosm, which you can see a diagram of here. This is basically, we can put up to 4 pairs in here. And then we have these resource zones. And so this is a picture of what volcosm looks like. We can control the light cycle, but during the night they get red light. Rodents can't see red, so this is essentially dark for them. And then inside of each one of these little resource zones, we have food. The water's in the middle here. We have a little video camera. They can play on a running wheel. And then we have a little device with which we can capture their vocalizations. They make these ultrasonic vocalizations. And in addition to that, on the sides of the room, we have these antennas. And these antennas are important because they allow us to actually track with real-time information where the voles are in the room. So this is one of the primary things that we're excited about, because what this antenna detects is an ultra-wideband device, which you can see here next to a quarter for scale. These are inexpensive and were initially developed so you could track packages going around warehouses. So we worked with a company to miniaturize this, and now what we can do is we can just put them in little hats on top of the vole heads, which have the added benefit of being able to label your animals so you know who's who. And then what we can do is we can actually release our animals into this indoor enclosure. And so what you see is a massively 80x sped up But you see the two females get introduced to this enclosure, and immediately you see like there are very big personality differences between these voles. Then we introduce some males, and then we let them just run around. We're doing unbiased discovery-based science to say, what do two voles do when you put them in a room? What do four voles do when you put them in a room? And we can take this and we can actually track them, and we can see where the animals spend the majority of their time. And so on the bottom here, you can see that this female really seems to like this zone, that she really avoids the center, probably finds it scary, and that overall the animals spend most of their time in the resource zones. Now, I like to think that basically my lab could start a reality show called something like The Secret Lives of Vole Wives, because we also like to spy on them in many other ways. And so what you can see on the top left here is basically the top-down view of the entire arena. This is, again, using AI to track these animals. Obviously not perfect yet in its tracking, but these are two animals that are just basically chasing each other around the enclosure. We can also voyeuristically watch what they're doing in the resource zones. Yes, we do get to see them mate. And then here, we can go ahead and eavesdrop on them. And so you don't have to listen. We've moved this. Normally they make these really high-pitched calls that you can't hear, so this has been moved into the human hearing range. In case you were wondering, this is what it sounds like when voles mate with each other. So we have just an absolutely huge amount of information on these animals, and we can validate and improve everything using these indoor arenas, and then we can basically move into nature. But how do we do this? How do we move animals into essentially real-ish nature? So the first step was to work with our landscape architecture team here at CU Boulder to identify someplace that we could actually build these enclosures. And then the campus architects actually had a really good idea, and she set us up with two professors in architecture and engineering, Marcel Delange and Jared Arp, who set up a capstone course for students in architecture, environmental design, etc. last spring. And those students actually designed our field research station. They designed it, they presented it to the review board for the entire University of Colorado, which is no small undertaking. They got approval, and then they actually started making a research station for us. Now, something like 3/4 of this video is just them digging, and I like to think that this is basically life imitating science, because what we're about to have happen is a whole bunch of voles digging in the ground. But this was, I think, an incredibly unique and powerful educational experience for these students, because as much as we might try to build laboratories and things, there is nothing that replaces a project done from beginning to end that's not something that's ever been done before. The other thing I really liked about this is there's this tendency for scientists and architects to not talk to each other when we develop scientific infrastructure. And this was pretty funny because there were a lot of design plans that they brought to us and we were like, that's never going to work. And so we really are winding up with a pretty amazing facility. So this is what it looks like. This picture is from July. Now, this is our research station. This is actually nearly complete. The roof went on today, and we're just waiting for the electrical. The enclosures, so we have two enclosures. We have the Alpha and the Bravo enclosures, but I will also say, if you would like to make a donation, I would be more than happy to name an enclosure after you. But there's also many other opportunities for how you can be involved in this moving forward. So these enclosures are 75 by 75 feet. And what we did is we used prairie dog fencing. So prairie dog fencing is used all the time in Colorado to keep prairie dogs out of areas that we don't want them. And in this case, it works equally well for keeping prairie voles in areas that we do want them. And then this is covered by netting, and then you can see that there's these posts sort of scattered throughout. The netting is just so that we don't lose voles to aerial predation. So each of those poles, we can go ahead and we can put one of those ultra-wideband antennas. This is what they look like when they're weatherized so that they can be outdoors. So we can track these animals now through this gigantic space, 24 hours a day, 7 days a week. And this is massively different than any study to date. Like, think about it. Before this, we were using live caps where we might capture an animal once every few days. This gives us continuous monitoring. We can go ahead and eavesdrop on them using that microphone to collect the ultrasonic vocalizations in the resource zones. And then sort of what I think is the most exciting is that we can take that work we did in dopamine, and now we can do it outdoors, because this device does the exact same thing, but it's wireless. So we don't have to have that fiber optic and big devices. We can literally do this in a field setting. So we have a lot of plans. The hardest part is probably choosing where to start. But what we're going to do with these sort of future Volcosm and Volterra is, first of all, take all of those things that I showed you today and ask, is this actually true in the real world? The second, though, is that just like humans, prairie voles do cheat on each other. So even though they bond, they've been known to be unfaithful, and this allows us to ask what variables actually contribute to the decision to either cheat on or divorce a partner. And along those lines, now we can ask what happens when love is lost in a much more naturalistic context. Because now we have communities of voles, and if one loses a partner, we can ask, what do they do? Do they give up their territory? Do they find a new partner? Are there different trajectories for different voles? And this is incredibly important because people grieve in different ways, and this may give us some insight into these different trajectories of grieving in humans. Okay, so now I'm going to do what I do at the end of every class, and I'm going to tell you what I hope you learned, which is, first of all, that voles provide this way for us to study this uniquely human behavior. I guess it's not uniquely human because they also show it. But the point here is that there is a richness of diversity in our world around us. Animals do amazing things behaviorally and otherwise, and by focusing exclusively on laboratory mice, we're really sort of putting blinders on ourselves because there's a whole suite of things that we can't study. Prairie voles give us the opportunity to study bonding, one such thing that we can't study in mice. Love is just chemicals and hormones. So I get asked occasionally by members of the popular press, is it just hormones? Now, as a biologist, I'm thinking to myself, is it just hormones? It's amazing that it's hormones. How do we have an incredibly complex biological system where you can have one molecule that can elicit this incredible complexity of behavior? And how does this actually happen? We really need, as social neuroscientists, to embrace the idea that we need to look at both individuals simultaneously if we really want to understand social behavior. Straightforward, obvious, not often done. And the last is that we need to study brains in the wild. And so these are the goals of my laboratory moving forward. So I can't end without acknowledging the people who actually do this work. So I have an absolutely incredible laboratory here at CU Boulder with people in it that work from everything from DNA all the way up to engineering and behavior, as well as my lab manager who basically keeps everything going. And so I'd be happy to take any questions.